Hello everyone. Chapter 4 is going to be a lot of review over some of the things that I know you learned in your prereq about eukaryotic cells like ours and the organelles and how they function. And then we'll look at the eukaryotic cells that are important pathogens. Recall in Chapter 1 we just introduced this idea of prokaryotes versus eukaryotes. We're going to focus on the eukaryotes today. We studied the bacteria, the prokaryotes, back in Chapter 3, so keep in mind those structures and the size of those cells dramatically smaller than the eukaryotic cells, and the main difference, of course, being the presence of a true nucleus or nuclear envelope. So keep in mind those prokaryotic cells or bacteria are very, very simple. They have similar structures to some of the eukaryotic structures we're going to talk about today, like the cell membrane. Some eukaryotes have cell walls. The glycocalyx, we'll see that on some eukaryotic cells as well. But because prokaryotes do not have a true nucleus, they still need a place to put their DNA, their chromosomes. So instead of having it wrapped up in a membrane, they just kind of push it to one side of the cell where they have an area where it's all kind of condensed in a little area called the nucleoid. They, of course, have cytoplasm and ribosomes. We talked about those in Chapter 3 as well. They have a slightly different kind of ribosome than most of the eukaryotic ones. They may or may not have flagella, but they may also have these things called inclusions. And inclusions may look like organelles, but they're not membrane bound, they're not separated in any way, and they're usually just collections of things. Sometimes it's a collection of nutrients. If it's a bacteria that needs to live deep in the ocean, that inclusion might be made of magnetite or something that draws it down toward the bottom of the ocean. So just keep in mind they're incredibly simple compared to the eukaryotic cells. So we're going to be reviewing each of these organelles here in just a moment, but just keep in mind again the relative size here. This is not to scale, clearly. The eukaryotes are going to be much bigger. Usually the range of eukaryotic cells is going to be anywhere from 2 to 100 micrometers, but we mentioned back in Chapter 2 when we were looking at the metric system and the size classes, we mentioned them getting up to at least 4 millimeters. So there's a wide variety of sizes in this group of eukaryotes that we're going to see today. Bacteria, on the other hand, they're going to be in the range of anywhere from 200 nanometers to 750 from 750 micrometers up to about 200 nanometers. So we're talking on the scale of 100 to 1,000 times different in size. So keep that in mind. So we introduced the major groups back in Chapter 1. We're going to look a little bit closer at some of the structures that differentiates these guys. Cell walls. So we said some eukaryotes have cell walls. Our cells don't, but microorganisms may. And so just like any organism that has a cell wall, its primary function is to protect it from osmosis, right? We're talking about organisms that live in aquatic environments. If it's a hypotonic environment, that water is going to want to rush into the cell, and that membrane is flexible, but it doesn't stretch, right? It's going to burst if it has too much water enter the cell. So that's really the primary function of cell walls. But it also gives the cells some shape, some support. But what they're made of depends on who you're talking about, right? So in the fungi, we have things like chitin and cellulose and really a broad variety of different kinds of carbohydrates, what they call mixed glycans. The algae, of course, we talked about these as well, the diatoms that have silica that make them very symmetrical and glass-like. We have carrageenan. We have also the substance that's extracted from a certain kind of algae that makes agar that we use in our culture media tubes. So we talked about that in Chapter 2 as well. But keep in mind, protozoa and helminths, the parasitic worms we're going to talk about, none of those have cell walls. So again, remember that protozoa includes amoeba, and they need a flexible membrane to move around. So that might help you remember that there are no protozoa that have cell walls, and same thing with helminths. Now, very generally, if you look at the structure, it's going to be in the same positions as we saw in the bacterial cell walls. So obviously the cell membrane, your phospholipid bilayer, is deep of these 3 layers. The cell wall, depending on what it's made of, is going to be superficial to that membrane. And then if there's a glycocalyx, it would be superficial yet to that cell wall. So picture that sort of layering as the protective coverings of these cells. So let's kind of walk through the organelles that we've studied in either A&P or biology, whichever prereq you had. Obviously, eukaryotes have a nucleus. It's protected by the nuclear envelope, which has pores so that the RNA and messenger RNA can leave the nucleus. But remember, there's also a little dense area called the nucleolus. This is where ribosomes are made, and then they're exported through those nuclear pores as well out into the cytoplasm. We're going to skip over lysosomes here because we're going to talk about them in a little bit more detail next. Mitochondria, of course, are the powerhouse of the cell. That's where we make ATP. Centrioles are involved in cell division. Those are the little anchors that we pull towards when we're doing either mitosis or binary fission, depending on which organism you're talking about. Then we move down here to the endoplasmic reticulum. Remember, we have two kinds of we have rough ER, which had the ribosomes, and that's where we make proteins. And we have the smooth ER, which is where we make either lipids or carbohydrates. And then once those things are made, they can be transported in little vesicles over to the Golgi apparatus, where then they can get modified and packaged depending on what substance we're talking about. Some of those will get modified and packaged into things like lysosomes, and some will get packaged into secretory vesicles for secretion of the substances. We mentioned the cell membrane is very similar to the bacterial one that we just talked about. The cytoskeleton here, again, because some eukaryotes don't have cell walls, that cytoskeleton is really largely responsible for the shape of the cell. But this is really a very dynamic set of protein rods moving through the cytoplasm that allows these vesicles to move around the cell. It gives the cell something to move the flagella if it had one, things like that. The cilia, of course, are going to be on the surface to help move things or move the whole cell. And of course, ribosomes, they may be free-floating or they may be attached to that rough endoplasmic reticulum. Because lysosomes are really key to a lot of the diseases we're going to talk about, we want to spend a little bit more time on this organelle. So remember, these are membrane-bound packages of digestive enzymes, and the enzymes themselves are called lysozymes. So realize that root word or that prefix lys means to break or cut. So obviously it's going to be a very powerful digestive enzyme for these cells that contain them. So what happens if this were an amoeba, of course, it would use those lysosomes to break down its nutrients. But where we want to focus on this organelle is really kind of going back into the human body where phagocytes do their job. So what happens is a phagocyte, when it comes in contact with something that needs to be consumed, it will package it up into a membrane-bound vesicle called a phagosome. Now the phagosome and the lysosome, which is in the cytoplasm just pre-packaged, waiting to be used, these two vesicles will then travel down that cytoskeleton and merge together. So again, why we put things in membrane-bound vesicles is so that we can use the ability of that membrane to fuse and seal and combine these. And once they're in there, once they have fused together, now they're called a phagolysosome. And so this is now where the lysosomes can actually, or the lysozymes, have contact with that bacteria or that whatever was consumed, and they can chop it up, break it up, and destroy it, right? So lysosomes are really important for phagocytosis. Since phagocytosis is one of the major defenses of the human immune system, there are some pathogens that actually use that interfere with this process, right? And so we talk about virulence factors. If there's something that they have that can get around some aspect of our immune system, then that's a way that they can cause disease. So this is going to be one of those virulence factors we see in quite a few bacterial infections. For example, tuberculosis. That bacteria, when it's back up here in the phagosome form or stage, it will actually release chemicals from that vesicle that will travel and block the cytoskeleton so that that lysosome can't come in contact with the phagosome. And so now essentially what you have is a happy little home for that tuberculosis bacteria. he's now inside of a phagocyte. There's no other part of the immune system that's ever going to come looking for him because he's inside of the phagocyte. And when you take the lysosome out of commission, you basically make that phagocyte useless, right? That's the only weapon it has, essentially. And so again, these are really key organelles. I want to make sure you understand how those work because they're going to be a common thing that we see with several diseases. In fact, the virulence factor is usually going to be referred to as a fusion inhibitor. And what it's inhibiting is the fusion of a phagosome to a lysosome. And so this is a nice summary table. We're going to talk about viruses, this far right column here. We're going to talk about viruses in the next chapter. So don't worry too much about those details at this point, but this is a really good summary table. So you can look at things up here like the true nucleus, the nuclear envelope, obviously absent in the bacteria and eukarya, but present in the eukaryotes. Some of the things they have in common would be things like binary fission. Some eukaryotes, like Paramecium, are going to go through binary fission, which is, it looks very similar to mitosis. They basically clone themselves and then split down the middle. We don't use the word mitosis when we're talking about bacterial cells. We would use the word binary fission. We have things like the organelles we just talked about. So obviously Golgi and endoplasmic reticulum, those are going to be absent in bacterial cells. They don't have organelles, but they do have ribosomes, right? They're just a slightly different type. Obviously, bacteria don't have mitochondria. They're organelle as well. In fact, what we do in the mitochondria, again, is make ATP. So bacteria, since they don't have a mitochondria, and you need a membrane to conduct the final stages of making ATP, what we call the electron transport chain, that's what's happening in the mitochondria of eukaryotic cells. Since bacteria don't have that, they actually use their cell membrane to do ATP production. So we'll see that when we get to the metabolism chapter. Some bacteria have pigments that can allow them to be photosynthetic. Of course, the algae would be the positive over here for the chloroplasts and pigments in eukaryotic cells. And keep in mind, we met the flagella and cilia in the last, in chapter 3. But keep in mind, remember that bacteria does not have cilia, right? Those cilia are huge compared to a bacteria, so there's no way that they would have a cilia on them. What they might have is lots and lots of flagella or fimbriae or some other extension that might confuse you about cilia. So just keep in mind these characteristics. Bacteria can have a flagella, but they can't have cilia. Eukaryotes can have one or both. The cell walls, cell membranes, those are going to be very similar between the two groups if the cell wall is present. And then glycocalyx. Even eukaryotes have some glycocalyces. And so here again you have, when they say the general sizes here, where some of those eukaryotes get much, much bigger, up to about 4 millimeters, but that's fairly rare. Most of them are in this general size class. So fungi, this is really a review from chapter 1. We said they can be multicellular in the mold and mushroom family. They can be unicellular in the form of yeast. Yeast are going to be the ones we consider most in this class because those are going to be the pathogenic forms. And so what makes fungi unique is that they are what we call heterotrophic in the sense that they have to get their food from outside sources as opposed to autotrophic, right? If something is photosynthetic, they can make their own food. So if they're heterotrophs, they have to absorb it from someone else. And so instead of consuming it like an amoeba would, they're going to actually just sort of dissolve it with enzymes and then absorb the nutrients. And so there's two general forms that we think of with this nutritional categories, what we call saprobic. Sometimes they call this saprophytic, is another word for that. But these would be the things you think of, sort of the fungal, the types of fungi that you would see on the forest floor, like mushrooms or molds growing on the surface of a water body, edges and things like that. These would be the things that are actually extracting nutrients from dead animals and plants. The ones that we're going to be worried about are the yeast forms, which would be considered parasitic because they're actually going to be inside of our bodies or inside of an animal body host, right? So these would be the yeast, parasitic forms of fungi. We also mentioned some uses for fungi besides just their functions out in nature as decomposers. We take advantage of some species of fungi. So we said foods, things like cheese, bread, alcohol, things like that. But the one I didn't mention was something called corn. This is one of those fungi that's used mostly in Europe and other parts of the world in place of meat, right? So there's a lot of meat alternatives, but there's a type of fungi that they grow that they literally, if you go to a McDonald's in some parts of the world, you can get corn nuggets instead of chicken nuggets, and they basically have the same taste and texture. Although there's been a lot of problems with allergic reactions to some of those, so I'm not sure how popular they are anymore, but there was a time where that's one of the ways we used fungi. Antibiotics we talked about, most of them are going to be soil residents that produce chemicals that we take advantage of and make antibiotics out of. When we say drugs, we mean both some pharmaceuticals, but also recreational drugs, right? So when people talk about magic mushrooms or shrooms, you're talking about two particular kinds of mushrooms that are secrete toxins or contain toxins that can affect the human nervous system, right? So that's what they're talking about there. And then, of course, we talked about the fecal waste management, and biofuels are some really interesting ways that we use fungi industrially or just marketable products. Let's move on to the protozoa. Again, we met these guys in chapter 1. Just in general, we group them by how they get around. So we have the amoeboid group. They use their pseudopods where they just kind of push that membrane out and then the rest of the cell just kind of oozes in behind it. We have the ciliates like Paramecium, and we have the flagellates, which are things like trypanosomes. Again, they're going to live mostly in aquatic systems, or they're going to require an animal host of some sort. And protozoan have a fairly simple life cycle. They have two main stages that they can exist in. Like the kind of what we call a trophozoite stage is where they're active and they're actually moving around, they're feeding, they're reproducing, things like that. But if the conditions that they live in, remember these all require some moisture. So if the conditions get harsh, if the water body they live in dries up, or the temperatures get too high, or nutrients are lacking, they will hide out in sort of a really protective coating called a cyst. So this is very similar to what bacteria do when they make endospores, but we would call them cysts in a protozoan. And then that cyst can literally, if it's in a water body, it can just settle down into the sediments of that water body. And then when the temperatures get better, the moisture comes back, they can just do what we call excyst, and they basically bust out of that cyst and then they're reactivated basically. So they have a very successful life cycle, but again, they require water. So they have this sort of protective stage. Now, keep in mind, not all protozoan can do this, right? So we have a couple of diseases we'll talk about in humans, like giardiasis. That one can encyst, and in fact, that's the major virulence factor, the fact that they can hide out and then come back to life basically when the conditions get better. But we have other ones that are the only place they are is in the human body. They don't tend to make cysts, so you have to have direct contact. So there's a disease called trick or trichomoniasis, which is a sexually transmitted disease, and so it has to go from host to host because they don't have this sort of protective cyst stage. So by no means do you need to memorize these scientific names here, but we're going to be meeting some of these diseases along the way. Things like the brain-eating amoeba, right? The Naegleria species. That's an interesting one we'll talk about when we get to the nervous system, which is, I believe, chapter 17. We just mentioned giardiasis. That'll be a digestive system disease. Trichomoniasis, reproductive system. Leishmaniasis, we'll see that in the skin chapter. So there's a lot of interesting diseases. What we call trypanosomiasis is sometimes referred to as Chagas disease. So we'll see that when we get to the cardiovascular disease chapter. Malaria, also cardiovascular, systemic infections. toxoplasmosis. So some of these are going to be interesting diseases we talk about later on. And then the last two here are both digestive diseases. So again, keep in mind you're not responsible for memorizing scientific names, but as we are introduced to these, if you want to at least kind of start recognizing their names and recognize a lot of these scientific names, they kind of tell you what they do, right? Toxoplasma causes toxoplasmosis. Cryptosporidium causes cryptosporidiosis. So some of them kind of give themselves away in that scientific name. It's the ones that don't really have an obvious connection that you might want to spend some more time with, like malaria, right? Plasmodium, it does not suggest malaria, and there's only one species that has malaria in the name. So, in fact, Plasmodium falciparum is actually the most common type, so, or the most virulent form anyway. Now let's move on to the helminths. So the macroscopic form is usually the adult stage. Keep in mind when you have a parasitic worm infestation is the proper word for that. We wouldn't call it an infection, although sometimes just in practice those words get exchanged, but we would say infestation for helminths or insect infestation. So things like scabies would be an infestation, or bed bugs would be an infestation. And any of these diseases we're about to talk about that are worm diseases would be infestations. So there's two main groups, what we call flatworms, which includes the tapeworms and flukes. And these guys are going to be very thin bodied organisms, and they have segmented bodies. So we're going to see, we'll look at their anatomy here momentarily. Roundworms, there's actually one of the roundworms actually is the name roundworm, but the group is called roundworms. So pinworms belong to the roundworm family, and that would be the nematodes. But these guys are going to be long cylindrical bodies that don't have segments. So that's generally how they're defined, and if you looked at them under the microscope, those characteristics could help you distinguish them. So let's look at the tapeworms, right? This is a classic flatworm. And so these organisms don't have their own digestive system. So they literally attach themselves to a host by way of this scolex, which is basically a little head with some sort of attachment piece, like suckers and hooks and suckers to attach to the host digestive system. And then basically the cuticle, which is the skin of the rest of the organism, is just able to absorb the nutrients from the host that it's living in. The rest of the body are the segments, which are called proglottids, and these are essentially nothing but gonad. But this kind of organism is unique in the sense, or not unique, but it's has both male and female gonads present, and so we call that monoecious. Right, in humans, you might want to be tempted to use the word hermaphroditic, right? We have both types of reproductive structures, but in tapeworms, we would call that monoecious. And so you can see each of these, if you could zoom in on one of those proglottids, what you would see is both a testis and a uterus, and so and ovaries. And so these can actually self-fertilize. They can cross-fertilize with other proglottids or other tapeworms that happen to be in the same host. So there's a lot of ways that they do this. And then basically as they mature, the proglottid farthest away from the scolex is the most mature, and they basically just drop off each of these segments. And so over the course of 24 hours, they may drop a dozen of those segments, and each one has, anywhere from 50 to 100,000 eggs in them, depending on what species you're talking about. Typically, we're talking either beef tapeworms or pork tapeworms, and so those, so it's easily transmitted from the host that's got one of these living in its gut. And so tapeworms have a life cycle that includes two critical pieces. What we call the definitive host is basically the host that contains the adult where the eggs are being produced, right? And so humans tend to be the definitive host for tapeworms. So the feces that come from the humans then get into the environment. If you live in a place where these are more common, it's because they live in a place where maybe they don't have proper sewage treatment. And so if an intermediate host, and again, there's preferred hosts for each of these species. So if for example, the pork tapeworm, the pig would be the intermediate host, and the intermediate host would be those that ingest the egg, and then the egg hatches in that particular host. And then once the egg hatches, it's the larvae that then migrate through the body, and typically will embed themselves in muscle tissue, but they can embed in pretty much any organ. How the life cycle completes is when they embed in muscle, and then humans eat that pork undercooked. Then the larvae then gets ingested, and it didn't get killed, and so it by cooking, so it basically enters into the next host, and this is the vicious cycle that occurs. Now, keep in mind, if humans were to ingest the egg stage rather than the larval stage, then if the egg hatches in the human body, that larvae would then migrate around its body and embed itself in tissues. And so it could embed in the cardiac muscle of the heart and cause heart problems. If it finds its way to the brain, it can embed in there, and you can have serious complications if you happen to be a human who is in the intermediate host stage. Now, fortunately, humans tend to avoid feces much more readily than livestock, perhaps, that are just, eating their food right off the ground. And so it's usually not a problem in that regard. But again, when you have places where people don't have proper sewage treatment, this can become a problem. Roundworms would include things like pinworms. And so in this case, again, they're long cylindrical worms that then when they get into the digestive tract, this one actually you have two separate species. So you have a male and a female. The female's much larger than the male typically. They find each other in the colon of the host that they happen to be infecting or infesting, excuse me. And they mate in the colon. How romantic, right? And then the female will then crawl out of the anus at night when the person is sleeping and is very still. That's sort of the trigger for the female to go out and basically go just out to the perianal region and lay the eggs, and then crawl back in and do it again. And so these eggs are consistently being placed on the surface of the body, and as they dry out, they become the skin becomes very itchy. So of course, where it itches, people scratch. So this is typically a problem with children who maybe don't have as good of hygiene as an adult might. And so this is a vicious cycle. When you get a child in a household with pinworms, it's very hard to get rid of them because they're constantly self-infecting themselves, right? They scratch, and then they don't wash their hands, and they end up ingesting the eggs, and they spread quite readily. The eggs can actually get in the bedding. They can get in the carpets and all kinds of, so it becomes really difficult to sometimes treat pinworm infections because they're sort of a vicious cycle. Now, some of the diseases we don't worry so much about in this part of the world, but they're actually the most common infections around the world. And usually it's in very poor parts of the world, in third world countries, developing countries. But very oftentimes where you have one, you have all three of these. So they're sometimes referred to as the unholy trinity. There's a good book that's referred to as the forgotten diseases, and these are all covered in that book. So how all of these work, they're called soil-transmitted helminths. So it's just what it sounds like. When you have skin that comes in contact with the soil, the larvae can burrow right through the skin, and when they get into the body, they will literally migrate around in the bloodstream for, up to 8 weeks. At some point, they will find their way to the lungs because the lungs are highly vascular. Once they get there, they will crawl into the alveoli, up through the respiratory tree. At the top of the trachea, they will get to the epiglottis, and they will climb over the epiglottis, find their way into the esophagus, and now they have been introduced into the digestive system. I know it sounds like a really convoluted way to get to the digestive system. Why didn't they just, their life cycle could be much simpler if they just got eaten, but that's not how it operates. So once it's in the digestive system, it literally, those larvae will mature by the time they get to the digestive system, and they will embed themselves in the lining and then essentially feed on the blood of the host bloodstream. And so obviously if they're consuming blood, they're destroying blood cells, you're going to have blood loss. So one of the most detrimental things about these infections is iron deficiency anemia, right? And so the person that's got the infection can then produce thousands of eggs in their fecal waste every day. So again, this becomes another vicious cycle. So now the eggs hatch and in the soil, and the larvae there in the soil can then reinfect the next person who comes in contact with it. But this 1 milliliter of blood loss a day might not sound like a lot, but keep in mind, you know, menstruating women who are probably much more well nourished than some of these children in this part of the world. A menstruating woman will lose about 0.6 milliliters a day, but she will only lose that for about 5 to 7 days. And even women who are fully nourished get anemic when with a 5-day period where they're losing some blood. So now picture a malnourished child losing a milliliter of blood a day, about 1.5 times what that woman is losing. But it happens daily for their entire lifetime. And so you're talking about poor growth and development stages. There's all kinds of other health consequences for being in that condition. So here are the organisms we're talking about: roundworms, whipworms, and hookworms. Now, there are different varieties of these that affect pets. So you might be thinking, oh yeah, my dog gets treated for hookworms, but these are different species. These are the ones that can affect human beings. And so they each, they can all cause iron deficiency anemia to some point and have a major impact on growth and development. But they each also have other problems. So roundworms, because they can become so large, they can literally obstruct the bowel. So when you see pictures of kids that look terribly malnourished, but they've got those big bulging bellies, there's two things that could be causing that. It could be roundworm infestation, or it could be ascites, which is basically they're so protein deficient that there's no albumin in their bloodstream. And so they're the only place where there's enough protein in their body is basically their core muscles. And so the fluid in the body is sort of accumulated there. Whipworms, they really do look like little whips when you can look at them microscopically. They tend to damage the colon so dramatically that they can have colitis, or rectal prolapse can occur when you have a whipworm infection that doesn't get treated. Hookworms are notorious for for causing really severe iron deficiency anemia. And you can sometimes see them when they're in those initial stages, when they're migrating through the skin and through the tissue. So you might think, well, there's an easy fix for this. Just give them all flip-flops, give them all shoes. It should be an easy fix. Unfortunately, that's not the case, because you're talking about places in the world where their feet are not the only contact that they have with the soil. They may work in the fields with their bare hands. They may sleep on a dirt floor. So we're talking about parts of the world where shoes are a luxury, along with many other kinds of luxuries. And so if you ever get the opportunity to do what they say, the classic example is deworming orphans in Africa, it's because in places where this is a problem, these children have to get treated every year. So there's no vaccine, there's no immunity formed once you've had an infection. So it needs to be constantly treated. And so if they're children that actually go to school, the teachers usually administer those drugs once a year, and they know they're getting regular medication. But their younger siblings at home may not be getting that medication. And so it's a vicious cycle in these parts of the world. So again, we're talking about other parts of the world, not necessarily the United States, but again, this is one of the most common groups of infections globally, and the majority of people that live in that part of the world have had to deal with it at some point in life. So here are a handful of these helminth diseases. There are more than 50 diseases caused by helminths that are going to affect human beings. We'll talk about a few of these later in the semester as well, for example, in the digestive system. But we'll mention one here that I don't think we'll be able to talk about later, is the river blindness. This is a helminth disease that gets transmitted through black flies, which would be very similar to what we have here as deer flies, right? These are biting flies, and they're also swarming flies. So you're talking about a place in the world where near riverbeds where you have these flies, they swarm, and if you were to get caught up in a swarm of these flies, you might come out with 200 bites in one exposure. And so they transmit the larvae when they bite the host. The larvae then migrate around the body through the bloodstream, and they find their way to the eye, and they will hatch, or the larvae will then enter the retina through the nervous system or through the bloodstream, and as the larvae accumulate in the back of the eye, the physician you can actually look in through the eye and see them swimming around in the vitreous humor of the eye. And so what happens is over time, it acts a lot like glaucoma, where you have too much pressure in the back of the eye. It puts pressure on the retina, it compresses the blood vessels, and eventually will destroy those neurons of the retina and cause blindness. And so it's really unfortunate because this is another one where there's actually very effective treatments. In fact, Merck, this is one of the few nice things I can say about pharmaceutical companies. The drug ivermectin is very effective. And they actually, back in the 80s, donated a lifetime supply, or agreed to donate lifetime supplies of this drug for this part of the world to try to deal with the blindness cases. And so the problem is the drug doesn't kill the adults, and so it needs to be treated every single year. And it's a real problem in parts of the world where there's war and people don't have access to the medical centers and things like that. So, this drug has been donated for free to these parts of the world 40 years ago, and it's still a problem today. So it's really unfortunate. So there's a lot of places in Africa that there would be really rich farming areas if it wasn't for diseases like this, where people just can't live because they're, of diseases like this. We're going to see this disease, schistosomiasis, which is the fluke. This is another one where sometimes they call that disease the swimmer's itch, because if you swim in a water body where you have excess nutrients, there's going to be a lot of snails, and the snails transmit this fluke. And when the larvae gets into the body, they'll migrate around. When they mature, they'll lay eggs, and if those eggs get lodged into places like the kidney, they'll calcify, and they'll basically lead to renal failure. So it's some really interesting diseases that come from these parasitic worms. Obviously, the highest rates of these types of diseases are going to be in tropical areas and places where they don't have proper sewage treatment, things like that. These are very successful pathogens. They lay millions of eggs every day. So if you have a place where you have sort of that vicious cycle of not being able to get away from the exposure, then it's a problem. So, all right, that's it.